So for those of you who haven't met me yet, I'm Arthur Lander. I come here from UC Irvine in California, where it is now somewhere around midnight. So I hope I remain standing through my talk. We'll see how that goes. Anyway, I want to thank the organizers for inviting me here. This has been fun. It's been great to meet you. And as you'll see, I'm very passionate about systems biology. I really think it is where biology is going, that if any of you do biology in the future, that you will be doing systems biology, whether you like it or not. So you can pat yourself on the back for having attended this course. It was the right decision. Okay, so before I get started talking about my topic, I'm actually going to put up some quotes here that were motivated, that have nothing to do with my topic today, but they were motivated by what Rob ended with in his talk. He talked a little bit about, hold on, there we go. He talked about sort of evolutionary just-so stories and why we shouldn't tell them. So I'm going to try to be a bit of a contrarian here. I have three quotes for you. One is from Francois Jacob, remember, Jacob and Monod? Okay. One is from Stephen Jay Gould, evolutionary theorist, and one is from Per Bach, the late physicist. And they each say something that's consistent with the other. Jacob says, living organisms are creations of history. They're not a perfect product of engineering. They're a patchwork of odd sets pieced together opportunistically. From opportunism of natural selection reflects the very nature of a historical process full of contingency or randomness, accident. Or Stephen Jay Gould says, if one small lineage of fishes hadn't evolved fins capable of bearing weight on land, terrestrial vertebrates like us would never have arisen. We're glorious accidents, unpredictable process, no drive to complexity. And then Per Bach, in the typical modesty of the physicist, titles his book How Nature Works, begins by insulting his audience in the soft sciences, that means biology, where contingency is pervasive, detailed long-term prediction is impossible. A science of evolutionary biology, for example, cannot explain why there are humans and why there are elephants. So I think we all get the point that each of these people are making, right? But I would argue that actually it's the sort of slavish devotion to this idea that there's so much randomness underlying what's out there in biology that we can't even begin to ask why things are the way they are. I think that's one of the reasons why the sort of molecular biology movement has failed, and systems biology has come along to try to take a different view of biology. Because I think that one of the distinctions between the systems biologist and the sort of previous molecular biologist is we're really not afraid to ask why. We understand that sometimes we will tell evolutionary just-so stories and they'll be wrong. But you know, just as we heard from Ted about Alan Turing's great paper, right? It said, yep, what we're going to tell is going to be false, but it's still going to be useful anyway, right? And so the point is trying to figure out what's the likely purpose of something in a biological system, right, is not a sterile exercise. It's actually critical to understanding biology, because ultimately biology is about fitness. It is about function. And so in my talks, I'm definitely going to take a very functional point of view. And we can debate later this issue of, where is the right balance to strike between sort of focusing on mechanism and focusing on function. So I just thought I'd put that up as a kind of contextual piece for you to think about as we talk about other topics. Okay, so I'm going to talk today about pattern formation. You've already heard some things in pattern formation from Ted, so that actually helps me because I'll be able to allude to some of what he was talking about. But in a break from a lot of what you've heard so far, just about everything I'm going to tell you about is about multicellular organisms, sometimes even big multicellular organisms like us. And so a lot of the issues that I'm going to be deal with controlling the processes that go into developing a multicellular organism. And so that's really the context in which I'm going to talk about pattern formation today. But I thought a good place to begin is just with definitions. So what is pattern? What do we mean when we talk about pattern? Right? So people think, okay, we mean stripes or we mean spots, or, but can we get a rigorous definition? So the best definition I can think of is some behavior that's stable in time And usually the behavior is cell differentiation that we're talking about. Cells become different kinds of cells that is regular in space, right? Has some sort of rules. So just a white noise in space is not a pattern. It's complex, but it's not a pattern. Pattern is something that follows some rules, and thereby we can divide things into whether it's following local rules, that is, the rules are defined every point in space relative to the points around it, or global rules, whether they're defined relative to some fixed landmarks. And we already heard a little bit about local rules, for example, in what a lot of the things that Ted talked about. And oftentimes it's called self-organized pattern, and oftentimes it's periodic or quasi-periodic, there are waves and so on. And one of the things that attracts so many people to biology is the beauty of the macroscopic patterns, right? In all the different organisms that we look at, whether you're interested in plants or animals or vertebrates or invertebrates. The pattern, the rules underlying them are just so cool. And from the quantitative standpoint, I mean, Ted already introduced this paper from Turing in '51 called The Chemical Basis of Morphogenesis, which, if anybody wants a copy, I actually have it on my computer. It's not too hard to find on the web. And it is a beautiful paper because he pointed out that what look like really, really complicated patterns can in fact arise through small variations on some very, very simple differential equations. I'm not going to go into a whole thing about Turing patterns. There's so much literature on it. If you want to learn about them, you can. They're an interesting mathematical problem because, in fact, there's not just one type of solution where you get a regular pattern in space, but there's 6 different sorts of solutions that can come out of these sort of Turing patterns where you have more than one diffusible substance capable of reacting. And even within this, the 6th case, which is the one that biologists mainly study, the stationary patterns, there's all kinds of visually distinctive patterns you can get based on small differences in the structure of the equations. And as Ted mentioned, Gierer and Meinhardt were responsible for sort of developing some of this theory on their own, and especially for popularizing it among biologists. And they introduced this formalism of activator-inhibitor or activator-depleter equations. And if you look at these are pretty simple equations. They say that a substance is able to diffuse. It's produced in a manner that depends upon its own concentration in a positive way, but is inhibited by the inhibitor. And it's degraded at a constant rate. And there's this little leak term here that's needed to get the system going if you don't give it a good initial state. And then the inhibitor follows the same rules, except it doesn't inhibit itself. And then you have 2 different diffusion coefficients. And that's really all you need in order to get patterns like this, where initialized from just white noise in the activator, they will spontaneously develop into zones of activator and inhibitor that look something like this. And there's lots of little apps and stuff on the web that you can get to play with Turing patterns if you like. In terms of solid evidence in support of this is a good theoretical model for these self-organizing patterns, I find that some of the nice recent evidence comes from Kondo in Japan, looking at the stripes. So this is the back end of a zebrafish. You can see a fin over here and a tail, and there are these 2 stripes. You can actually go and cut the skin out and make a wound, and you get rid of these pigment cell stripes. And if you look at the way it heals, the stripes don't heal by reforming the stripes, but rather there's a nearby stripe that sort of spits off a new stripelet, which then moves into the space. And meanwhile, these guys get a little bit fatter on their ends while that's going on. And if you model this as a Turing system, you get exactly that sort of behavior. This is just a model on the side here. Essentially, it's reaching a state of trying to regularize the spacing between things, right? And so as a result, it's much more likely to have this thing bend in than have those things move over. So these are the sorts of things that suggest that even if the model is formulated at this kind of high level with sort of fictitious activators and inhibitors, that fundamentally it's probably capturing something right about a lot of these biological patterns. Now, personally, I find for a lot of things that are quantitative in biology, I don't really get them as a take-home message unless I can think of some sort of metaphor or something to draw an analogy to that I understand better. And so, I've worked to sort of have ways to think about why Turing patterning works that I can connect to things I understand a little better. And so one way to think about the whole Turing patterning process is as a spatial generalization of the notion of a feedback oscillator. Okay, so remember, a feedback oscillator is something that feeds back negatively on itself, but there's a time delay, right? So it goes up, and then it feeds back and goes down, and then it goes up, and then it goes down, right? And that's what's shown here, right? Well, so if you just substitute space for time, right? If feeding back an opposing signal at a different point in time can produce oscillations in time, then feeding back an opposing signal at a different point in space might be able to produce oscillations in space. Well, how do you feed back at a different point in space? It just means the thing doing the feeding back has to spread at a different rate than the thing that's being fed back upon. And so it feeds back more over here than it does over there, and that produces oscillations in space. And that gives you one of the sort of criteria for Turing patterns is that the inhibitor must have a longer spatial scale than the activator, and that's actually true for them. So the other way to think about Turing patterns is as a spatial generalization of bistability. Everybody know what bistability is, right? They have two stable steady states. This is a typical example of a pair of differential equations that give you a bistable system, right? You can be at a low state or a high state, and which one you're at depends upon your initial conditions, right? Whether you're coming from this side or coming from that side. And so you can sort of think of a Turing pattern as having both high and low states in the same place, and you initialize from different positions at different locations. So you alternatively go between high and low states in these patterns. And that explains another thing about Turing patterns, which is that you need to have nonlinearity in the activation or the activation and the inhibition. Otherwise, instead of getting bistability, you get monostability. And the same is also true in Turing patterns. It has to be nonlinear. It has to be greater than linear in order to get a pattern at all. Anyway, so those are sort of two conceptual ways to think about the basis of Turing patterns. Okay. But there's a lot of practical problems with the whole theory of Turing patterns when you try to use them to explain all of biology. And as Ted alluded to this fact, is there's been a period of about 30 years when biologists kind of fell out of love with Turing patterns because some people tried to use them to explain everything in biology, and they clearly didn't work. That's kind of changing now. I mean, people are falling a little back in love with them, but not because they're explaining everything, but rather because they're very specific things that they're explaining. But anyway, so one of the problems is that the theory is formulated at a very high level, right? It's just like activator, inhibitor. What does exactly that mean? It could mean anything. It doesn't even have to be a substance. It can be a mechanical effect and so on. And so experimental biologists hate that, right? It doesn't help them at all if the theory doesn't give them a clue what kind of thing they'll be looking for. There are also some inherent disadvantages that come from the fact that Turing patterning is about local rules, right? Which is that oftentimes you want pattern to respect global information. And the classic situation has to do with the scaling of pattern. So this is a fruit fly that was starved during its development. This is a well-fed fruit fly. And they're both perfectly normal fruit flies, except this one is smaller, right? All of it is smaller, proportionally smaller, including all the patterns. Right? All the patterns have been scaled down. And as you evolve organisms of different size, the patterns seem to scale on their own to fit the size. And at the end, I'll show you some other interesting examples of scaling. Well, the problem, question?
What happens to cell size among the two?
So that's really interesting. So it turns out there are cases in which scaling is largely a cell size effect, and then there are other cases in which it's largely a cell number effect, and then other cases in which it's a mixture of the two. And pattern seems to come out just fine in all those cases. So, in starvation, it's largely a cell size effect, I think. But there are many ways to make the cell number change, but keep the size constant, and then the pattern stays constant. So somehow pattern seems to be decoupled from both of those things. Anyway, so the point is, if you look at a Turing pattern, right, the parameters dictate the wavelength, right? So unless somehow the parameters are perfectly adjusted, and it's not a linear adjustment to match the size of things, then Turing patterns won't scale, right? So there's some trickiness there. And so consequently, a lot of people in biology who focus on pattern focus on this issue of global rules, right? How you get cells to respect information about how far they are from a particular location. And so, for example, in a fruit fly, the embryo has to be divided into segments of different classes of gene expression. And even though these are stripes, this isn't really a Turing pattern. It's not arising in order to sort of keep the distance between stripes the same. Although, I'll come back to that a little bit later. There's actually Turing-like aspects to it. But by and large, where gene expression occurs is a function of distance to an organizer, a point in space where something's happening that's providing positional information to the cells along the way. And the same is true for other patterns. This is the fly wing. It's actually homologous to our hand, or to our arm, I should say. And in this case, it has pattern, the location of these veins along the wing, and their distance from the center. And again, that pattern is determined by something that's coming out from this organizing region, which I'll show you in a moment. So to most biologists, the term morphogen is used to talk about this kind of patterning, even though Alan Turing defined the term first to talk about his kind of patterning. So there's a bit of a confusion in the literature. But most biologists, when you say, oh yeah, you're working on a morphogen gradient, you're talking about this sort of thing where there's an organizing region that produces something and it forms a gradient in space. There's all kinds of things those could be. And then cells turn on different patterns of gene expression depending upon their position relative to that source. Okay. And so in a sense, that's kind of like cheating, right? It's not a self-organizing pattern. The pattern isn't emerging out of randomness. Rather, you've already started with symmetry being broken, right? You know that there's something over here, and you're just organizing other cells at distances from it, right? And so you're kind of parlaying pattern into more pattern, but it turns out a lot of biology works that way. A lot of pattern is built on previous pattern, and so on. Now, morphogens form gradients, as we'll see, and that's how cells get their position and positional information. And so you've kind of already heard about gradients, and you will hear even more about gradients in this course in the context of chemotaxis. And so even though gradients are studied in both realms of biology, whether you're interested in chemotaxis or morphogen gradients, there are big differences in emphasis. Because in the case of chemotaxis, the gradient is something about the environment usually, which you can't really control. It's what it is. And so most of the focus is on how the cells have machinery to be able to detect all kinds of unpredictable gradients and do the best job they can in them. In the case of a morphogen gradient, it's kind of flipped around the other way, right? You can set up the morphogen gradient you like, and then the cells can just be stupid, right? They can just say, okay, I see that, I'll do this. So a lot of the focus really is on how you set up the right morphogen gradients in the first place, right? How you control them. So there are, differences in emphasis, but in both cases, gradients are associated with positional information. So one of the most studied, some would say overstudied, morphogen gradients in biology is the bicoid system. It involves this very early Drosophila embryo. And Drosophila embryos are kind of weird. They start out, like all embryos, as a single cell. But in their case, the nucleus divides 14 or so times without any cell divisions. So you get one big cell with 2 to the 14th nuclei in it. And then those nuclei all migrate to the edges, and then the cell membranes come in afterwards. It's kind of weird, but the consequence of that is during that first period, the syncytial blastoderm period it's called. During that first period, things can diffuse around inside the cytoplasm of this huge egg and get from nucleus to nucleus. So information can be transferred from one cell to another without actually having to leave any cells. It just kind of flows from one to another. So it's kind of an unusual way of developing, but that's how Drosophila do it. And so consequently, morphogens can be intracellular molecules in that case. Most of biology, morphogens are things that go outside cells and then move around from cell to cell. So bicoid is one of those intracellular morphogens because during the way the egg was manufactured, bicoid RNA was all placed at one end. You get a gradient of bicoid protein, and you can actually plot that gradient. It looks like a nice exponential shape. The other system that I'll mention has to do with the wing. I already showed you the nice pattern of the wing. Where that comes from is from a flat sheet of cells in the larva or maggot, which develops during larval life, sitting under the skin, and then later during pupation turns into a wing. So this is really the structure, the wing disc, that has to have the patterning imposed upon it. And so that disc is going to turn into this. And there are three morphogens that are involved in doing that. There's a morphogen called hedgehog. All the genes in Drosophila have silly names because they were named after phenotypes. Anyway, hedgehog is one morphogen. Decapentaplegic, or DPP, is another morphogen. And wingless is another morphogen. You can imagine the phenotype of that, right? No wing. Yeah, anyway. But these all have homologs in other organisms as well. Most of the world uses these terms. But anyway, we'll call them hedgehog, DPP, and wingless. And different aspects of the pattern of the wing are under the control of different morphogens. So hedgehog pretty much determines where these two longitudinal veins go. DPP determines where these two longitudinal and these cross veins go. And wingless patterns the bristles here on the outer margin of the wing. So what does a wing disc look like? I said it was a flat sheet. It's actually two sheets. I've turned it on the side and done a cross section. So there's a fat region called the columnar layer, and then the sheet sort of bends around and has another thin layer called the parapodial membrane on top. But this is the part that's patterned and actually gives rise to the wing blade. This parapodial part is eventually discarded. So essentially, it's a two-dimensional patterning problem. How do you impose on that sheet of cells seen here in cross-section? They're very long and skinny, by the way. How do you impose pattern?
Is that like 20 microns from bottom to top?
Bigger than that. It's more like 30 microns here. But each cell is only about 1.5 microns wide. So it's like a bunch of stacked straws. Okay, they're very, and we'll talk a little bit later why it would make sense that they would have this shape. And this distance from here to here is about 80 or 90 microns, 80 or 90, maybe 100 microns in that direction. So a couple hundred microns at most, and about 30 microns tall. So if we look from the top down on that, all right, so now we're looking at the top on the sheet of cells. Here's the sheet. And it already has an anterior-posterior identity because it came from the early embryo, which had segments, and the segments already had different gene expression in the anterior and posterior halves. That's true in the wing disc as well. The posterior half expresses a transcription factor called engrailed. Engrailed does two things. It activates the expression of the morphogen hedgehog and shuts down the expression of a necessary receptor for hedgehog. So therefore, even though they make the morphogen, these cells can't respond to it. The only cells who can respond are the ones next door. So this is what Ethan Beer calls a for export only signal. And so consequently, the cells next door see hedgehog that's diffused over, and they turn on the second morphogen, decapentaplegic, or DPP. So consequently, you get a stripe of expression of this morphogen. And then this morphogen spreads out and forms two gradients. And this is the best I can do drawing a gradient in PowerPoint. But anyway, DPP is declining and declining as it goes away from this stripe. And fortunately, we can visualize these gradients. For example, several labs have made fusions of DPP to green fluorescent protein and put that in the genome. And it's a little bright in here, so you can't see that well. But there's a gradient of little green dots coming out here, which is representative of these two DPP gradients going in the posterior and anterior directions. Anyway, what happens ultimately is that at least 3 different genes get expressed in different patterns based on where the cells sit in the DPP gradient. If you sit too far from DPP, you turn on Brinker. If you sit intermediate, you turn on OMB. And if you sit really close, you turn on... Who did that? There's poltergeists in here. Somebody turn the projector off by itself? Okay. Flies doesn't want me to talk about that. Okay, I'll start drawing it. Basically, oh, here we go again. Okay, I'll use my hands, all right? So you have these nested patterns of gene expression, and what happens is at the boundaries between patterns of gene expression, where one gene expression abuts another, right? The cells talk to each other. And that then induces other genes at those boundaries. And those are ultimately going to be what corresponds to where those veins are going to form. Okay, I see a light coming on. I see a light at the end of the tunnel. Yeah, there's some light coming out of it. There you go. Okay, it'll warm up. Let's hope that doesn't happen again. Anyway, so at the boundaries between gene expression domains, other genes are turned on, like these genes, like rhomboid, which is involved in epidermal growth factor signaling. You can see these stripes. This is going to be where vein 1, 2, 3, 4, and 5 form. There are 5 veins in Drosophila. There's more in other insects. And so eventually, it's these things that are somehow going to narrow down to being a very thin stripe, which is going to be these longitudinal veins. And that process, I'm not going to talk about today. That involves re-expressing DPP and another morphogen and using another mechanism of signaling. And actually, it has characteristics of a Turing pattern in forming these stripes in the very last stage. That happens during pupal development. But ultimately, that's kind of start to finish how you get from just one polarity cue in the wing, which is what's posterior and what's anterior, to making 5 veins that go down the wing. And okay, and then wingless is expressed in a different pattern. It's expressed in the dorsoventral boundary. So it's kind of like a Cartesian coordinate system, right? You have something that's expressed along the anterior-posterior boundary and patterns things in this direction. And then wingless is expressed along the dorsoventral boundary, and it patterns things in that direction. And just like DPP, there are targets that are turned on very close to wingless. Here's an example. Here's an example. And targets that are turned on farther away, like here, and here, and here, and so on. So again, it's that same sense of an organizing center and things turned on at a distance. Okay, and then there are a number of other systems, most of which I'm not going to get to talk about, but if you're interested in, there's a lot of literature on, how we specify what's our back and what's our belly. That's done with long-range morphogens. How you determine where your motor and sensory parts come out in your spinal cord. 5 fingers, right? Why 5? Why not 7? Why not 12? And so on. Patterning of the hindbrain. Our brains are segmented. Or even our vertebral bodies, right? These are another pattern formation system that uses long-range morphogens. Okay, so that's kind of an introduction to sort of morphogens and how they're used. All right, now how do these gradients form? This is something that people have been thinking about ever since they realized that information about position was being passed at long range. And the earliest ideas were very simple ideas of that came from understanding diffusion, that, if you have something that's produced at a source and is absorbed at a sink, if nothing else is going on, then you just, solve fixed laws, right? And you get a straight line, right? Because the second derivative is 0, right? So you get a straight line from source to sink. And so that was really the first idea most people had about how these morphogen gradients worked. Even though they didn't know anything about what the sink might be, right? The idea was somehow there was a distant sink. And one of the nice things about the source-sink model, and in fact, this is what Lewis Wolpert, who most biologists credit with having thought up the morphogen gradient idea, but in fact, it's a much older idea. But what he did do is he pointed out a useful thing about a source-sink model, which is that it automatically scales. Remember I said there's this issue with Turing patterns that they don't scale because they're local, not global. In this, if I just move the sink over here, right, then the line just changes its slope to accommodate the new location of the sink. So anything that's specified at certain positions will move out proportionally. Everything will scale beautifully. And Wolpert pointed out that was a neat thing about source-sink gradients. Now, one of the questions that came up early on, yeah?
Here, because actually there's another assumption that goes in.
Oh, yes, there is.
You actually pin the concentration.
Yes, there is, and I'll get to that. And I'll get to that, Boris. Okay, sorry. You didn't think I'd fall for that. Okay. But I just want to make a point about this issue of diffusion, because this is Francis Crick, right? Remember central dogma? There it is. There's the central dogma on the board, right? They had to take a picture of Crick with the central dogma behind him. Anyway, there was an interesting interchange that went on just after Wolpert's paper in 1969 pointing out morphogen gradients and this whole scaling issue. Wolpert said, actually in the abstract, I think it was, or the introduction of his paper, it has been a great surprise that most embryonic fields, that means the areas over which morphogens pattern, seem to involve distances of less than 100 cells and often less than 50. Okay? And so typically that means we're talking about distances of 100 microns or smaller. Most morphogen gradients act at less than 100 microns, which, if there's a reason for that, is pretty darn important, right? Because you've got to get your patterning done then while everything's very, very small, and that creates all kinds of potential problems. So as soon as he published that, Crick sat down and kind of did some back-of-the-envelope calculations. And in those days, if you were Francis Crick, your back-of-the-envelope calculations became Nature papers. And it did become a Nature paper in which he said, a simple order of magnitude calculation suggests that diffusion must be the mechanism by which morphogen gradients form because of what Lewis Wolpert said. So can anybody guess what it was that Crick pointed out? Okay, remember fixed law, diffusion, right? How long does it take something to diffuse a certain distance? How does that scale with distance? It scales with the square of distance. So Crick's point was development has almost only so much time to do its process. And his back-of-the-envelope calculation was to put in a few diffusion coefficients, a few sizes for development, how long development is supposed to take. And he said, that's why it's limited to about 100 microns, because to diffuse longer than that would just take too long. That was a Nature paper. And in fact, it is still cited as today as the reason why we believe morphogens diffuse is because of this range limitation. So it turned out that Crick was wrong, not surprisingly given the way I explained it, right? No, but the interesting thing is why he was wrong, because he was wrong on two accounts. And I think it's actually very instructive to sort of dissect a little bit why Crick was wrong, okay? So the first reason he was wrong was because one of his assumptions was unjustified. And the assumption was a constant rate of morphogen production, right? What if the rate of morphogen production were very high at the beginning and then very low later, right? Well, then you could get to steady state a whole lot faster. And in fact, engineers know this, and even pharmacologists know this. If you've ever been told if you have to get on some medication regime, on the first day you take 2 pills, right? Because it gets you up to steady state faster. So in fact, if you can produce the morphogen at a variable rate, you can get to steady state at arbitrary distances, at arbitrary times. There's no limitation whatsoever. So that's kind of interesting, right? Well, we'll come back to that. But he assumed the system was the simplest possible system, but then he used it to kind of derive constraints when biology can get around those constraints just by putting in the tiniest bit of complexity. The second is that morphogen gradients aren't source-sink gradients, right? I mean, this was just a convenient model for thinking about them. But most morphogen gradients are not linear, they're exponential, right? And this is actually a fit of the bicoid gradient as measured by fluorescence intensity using calibrated antibodies to bicoid. And you can see it's very nice on a log plot. It forms a beautiful straight line. So that's a real exponential gradient. In fact, this is true for other gradients. The data are never as pretty. This is the DPP gradient in the wing disk on the posterior compartment. And you can at least fit the average data. Makes me a little bit nervous, but you can fit the average data pretty nicely to an exponential in this case. It's certainly not linear. And when you think about it, exponential shape is not surprising, because if you take fixed equations, right, that the rate of change of the ligand over time, right, you have the diffusion term, you have a production term, which is presumably just at one end. But if you now put in a degradation term, so it's degraded at a uniform rate everywhere instead of just at a sink, well, that just solves the declining exponential. Okay, so the shapes of morphogen gradients are consistent with just diffusion with constant degradation.
Somebody brought up previously about effects of boundaries on simple diffusion, like diffusion. So in this case, don't boundaries play a role that you are diffusing some molecules across
Yes, they may play a role.
molecules at the boundaries. Something moving like this and it starts coming back from the other side. Wouldn't that play a role?
Absolutely. So this solution to that equation is assuming that the boundary is at infinity, right? And if you bring the boundary in from infinity, it's not a perfect exponential. Okay, but what's nice about this solution is, see this number here, lambda, the length constant or length scale. That also tells you the distance over which boundary effects occur. Okay, so if the boundary is more than several lambdas away from the origin, then the boundary effects are negligible. Okay? And in a lot of these, but not every system, a lot of these, the boundaries are quite far away. And so at one end, you can essentially treat it as though it's an infinite boundary. So, okay, good point. Yeah, this is just a spatially dependent. I mean, and typically you just have one region where you have production and nothing elsewhere, which is much more reasonable than a boundary condition of production, which gets a bit at Boris's comment that I cut him off because I'll show it later. Anyway, so it turns out you can take that same equation and solve it not in the steady state, but in the time domain. I'm not sure you could do it in 1970. Well, you probably could, but no one ever did. But eventually, people did sit down and solve it. And it's pretty ugly, but in fact, a very close approximation of this is just that the rate of approach to steady state is proportional to time, not proportional to distance, not distance squared, linearly with distance. And these are just some simulations showing you either how the gradient progresses over time or the fraction of the way to steady state at different positions. And you can plot that, and sure enough, it goes linearly. So Crick's whole reasoning that it would slow down, right, as you went farther away is just plain wrong, right? And it's wrong because it only applies to the source sink model, and it doesn't apply to a uniform decay model. So anyway, so be careful when you build simple models. They're good to explore how things might happen, but then when you say the constraints that come out of the models must apply to biology, be careful that those constraints aren't the function of your assumptions rather than a function of the process. Okay, so now many people have wondered, since we can't use Crick's argument that morphogen gradients formed by diffusion, maybe something other than diffusion is responsible for moving morphogens. And a lot of the sort of motivation behind that has come from people actually looking at these fluorescently tagged morphogens. One of the things you notice when you fluorescently tag DPP, so here's the end of the production region. So this is the gradient out here. And you see it's in these blobs. And if you look closely where the blobs are, they're actually inside cells. You don't see any DPP between the cells or above the cells. You see it inside the cells. And so that led people to suspect maybe DPP is moving inside the cells from one cell to another. And another piece of data that suggested that was the case is if you transiently block the ability of cells over here to take up morphogen into them, that there was like a shadow behind them of less morphogen for a while until it came around from that side. So these sort of transient shadows were also taken as evidence that maybe the morphogen is moving in and out of cells. And then somehow by the end of the first decade of the century, even Louis Wolpert was wondering whether morphogen gradients had anything to do with diffusion. He decided diffusion was too messy and too hard to control. And ultimately, this led to some groups trying to measure the rate of transport. And one of the tools we often use in biology to measure transport rates is called fluorescence recovery after photobleaching, or FRAP. And here's a case in which this was used on the DPP gradient. So how many people have heard of FRAP? A couple, all right. So basically, if you have a fluorescent molecule, you bleach it with a laser, so you now have a spot that's not fluorescent anymore, and you look at the rate at which fluorescence recovers, and that depends upon transport of fluorescent molecules into that spot. Okay, and so you get essentially a relaxation time, and maybe some molecules don't relax. That's another story, but you get relaxation time, and from that relaxation time, you try to infer a diffusion coefficient. And people who've done that in the DPP gradient get a diffusion coefficient a couple of orders of magnitude less than what you'd expect for a protein diffusing in, say, cytoplasm or intercellular space. And so then people have come up with all kinds of ideas, like, well, maybe it's going in and out of cells, and maybe it's going around cells, and maybe cells are sending Philopodia over and grabbing it. And so there's been a lot of confusion about how morphogens are transported. But it turns out when you look at the problem closely, you begin to wonder whether those conclusions drawn from photobleaching, or FRAP, are actually giving us the right picture. Because there's another technique called fluorescence correlation spectroscopy. How many people have heard of that one? A few. Okay. This is a very physics technique, so you should know it. I mean, essentially, what you're doing is you're illuminating one very small pixel, and you're looking at the noise in the signal coming back from that pixel. So the noise, the fluctuations, represents the statistical fluctuations in the number of molecules in that volume that are fluorescent. So as molecules wander in and out, you see a noise in the trace. So if you do autocorrelation on that noise, you can figure out the mean passage time to wander across a pixel. And that essentially gives you the diffusion coefficient. So if you do that with DPP in the wing disk, in the intercellular spaces, you see, in fact, that about half of it is moving at a speed of about 10 to 20 microns squared per second, just like you'd expect for a free diffusing molecule. And about another half of it looks like it's stuck to the plasma membrane. And so it has a diffusion coefficient a couple orders of magnitude lower than that. And so it's interesting that the same discrepancy between the number you get from FRAP and the number you get from FCS has arisen in other morphogen gradients. For example, in the bicoid morphogen gradient, if you do FRAP, you get that it spreads at a rate of 0.3 microns squared per second. If you do FCS, you get 7 microns squared per second. Right? So you get a big discrepancy for the two. The interesting thing about the bicoid gradient is that the value that you get from FRAP is actually too slow to explain how rapidly the gradient forms in vivo. This is a beautiful picture from Thomas Gregor, a movie actually showing the bicoid gradient forming with fluorescent bicoid over time. These pulses are, every time the nuclei divide, the bicoid goes out and then has to be pumped back in. And you can see this beautiful gradient form, right? And you can tell that the gradient basically takes about an hour to reach a steady state. But remember this picture for the simple exponential gradient from which you can calculate how quickly it's supposed to go to steady state. And if you put in the number 0.3 microns squared per second, you get it should take 6.4 hours to reach steady state instead of about an hour. So that suggests that that number is wrong. And so the question is, how could FRAP be giving wrong answers? And again, it's this issue of assumptions. There's a big assumption you make every time you do FRAP, which is that transport is the only process that's affecting this relaxation time. if there's anything going on in series with transport, it can affect the relaxation time. And in the case of DPP, if DPP is diffusing between cells, but then is being captured and taken up and stored, and only later being degraded, right? Then the relaxation time you measure with FRAP is going to measure this whole cascade here, and is essentially going to be determined by whatever is the slowest rate constant here, right? And it turns out that if you allow for this sort of model, you can fit exactly the data that were used to get a rate constant of 0.1 square micron per second with a diffusion coefficient of 20 square microns per second. So really, FRAP can't be used to infer the rate at which something's moving if there's other stuff happening as well, right? And it turns out that's just showing you the rate constant you'd get when there are other compartments. And it turns out in the DPP gradient, you can show that that's really what's going on, because you can do another trick called photoactivation, which is kind of like the flip side of FRAP. Instead of bleaching something fluorescent, you've engineered something that only becomes fluorescent after you've hit it with a laser. And so you can take you can make stripes on a DPP gradient where you suddenly make the DPP fluorescent, and then you watch it move, except it doesn't move, because 99% of the DPP is inside cells and it's going nowhere. It's all been stored, and it's the tiny amount that you can't see that's responsible for the whole morphogen gradient. Okay, so any questions about that part? All right, so basically what I'm saying is morphogen gradients, morphogens can diffuse between cells, but the morphogen you see isn't necessarily the stuff that's being transported. You can be seeing the stuff that's accumulated after transport and is hanging around inside the cells. And it may track the gradient of free morphogen, but then again, it may not. We'll come to that one in a minute. Okay. So that's just some fundamentals about morphogen gradients, their shapes, and how they form. And now I want to talk about the sort of nitty-gritty details of what's actually controlling the form of morphogen gradients in vivo. So first of all, I said that simple models of diffusion with uniform uptake are consistent with these exponential shapes that you tend to see for morphogen gradients. Also, those models predict some nice in vivo behaviors. For example, in the DPP gradient, we can manipulate things. We can take away one copy of the receptor. Receptor is called thick veins. Or we can overexpress the receptor. When we overexpress the receptor, the morphogen gradient shrinks in. When we take away one copy, the morphogen gradient expands out. Okay, why is that? So it's consistent with the fact that in the simple exponential model, this length constant, lambda, is equal to the square root of the diffusion coefficient divided by this uptake coefficient, or the coefficient of which, this rate constant for being removed from the diffusing pool. A lot of people call this a degradation rate constant. It's not. It's the rate constant of capture or being removed from the pool. And this, if receptors are responsible for removing the morphogen, which in most cases they are, then this will be proportional to morphogen, and so proportional to receptors. So more receptors, shorter length constant. Shorter length constant, the thing spreads farther. Okay, that makes sense? Right. In fact, here's just a nice little simulation. Here's the actual equations with the free morphogen, the bound morphogen, and the receptors. And here, if receptors go up, the thing moves like that. And if receptors go down, it spreads out again. Okay? So the data seem to fit that very nicely. And so at the end of the day, you're left feeling like a good molecular biologist with the sense that you can reduce everything to a nice linear pathway. Yeah? Well, it'll be a ways before I finish. So.
I was curious in the previous example, if you looked at a different species, kind of like Thomas did, because in that case, the real failing was when you changed species. In one species, the lambda, basically
right.
the lambda constant made sense.
Right.
If you had a larger fly, it
right.
worked out. So I'm kind of curious here,
has it?
Fortuitous.
If what's fortuitous?
Well, you're saying that the thing is consistent with the exponential and the profile being x over lambda. And
right.
I'm just saying that now, like the next thing I would be tempted by
It does scale, and I will show you pictures of it scaling. However, it's definitely, did this happen again? Now it's over here. Whoa! Hey, this is really confusing. Can I look at myself, looking at myself, looking at myself, looking at myself? I don't care. I'll point over here until you sort this out. Yeah, I'll show you that as well. I mean, it isn't a simple exponential, right? But let me build that up slowly because I'm starting to, right? So if you're a good molecular biologist, we're back here now. Okay, if you're doing this because I'm jet lagged, I know you're okay. If you're a good molecular biologist, you build a linear pathway and then you go home and say I'm done, right? And so we can do that at this point. In Grail turns on hedgehog. Hedgehog signals through patched. I have this because the more receptors you have, the less far it goes, right? So there's this kind of strange sort of negative effect here. Patch turns on DPP. DPP, same thing with its receptor. DPP receptors, when occupied, lead to the phosphorylation of the MAD protein. And that has effects on the Brinker gene and then on other genes and so on. It's all molecular biology from there on down, right? This is the simple version. Okay, let's start dealing with the realities. Reality number one, hedgehog controls its own receptor. So its own signal feeds back to upregulate its own receptor. And since its receptor shortens its range, hedgehog shortens its own range, which then shortens its own range, right? There's a kind of positive feedback going on here. We'll talk about that a little more in a moment. Second, the rate of production of receptors is not constant. It's affected by hedgehog, by engrailed, and by the morphogen itself, such that the more DPP you have, the fewer receptors you have. And consequently, the receptors are not uniform in space. The receptors are in a counter gradient, higher on one end than the other because engrailed turns them on, right? And so consequently, the gradient of phosphomad, the signal, doesn't look exactly like the gradient of DPP, but it goes up and then down and then up and then down, right? Okay, so you know right away this is not a simple exponential anymore. But it gets better because it turns out there's another molecule called DALI on the cell surface, which is a proteoglycan. It interacts with DPP, and somehow that stimulates the signal, so you get a much stronger signal when DALI is present. And DALI is controlled by DPP and hedgehog, but in a slightly different way because it's activated rather than inhibited, and so consequently the highest levels of DALI are here in the middle where hedgehog is acting. Remember, hedgehog's coming over from the posterior side. All right, but that's not enough because there's another morphogen in this system called GBB, which is what you get if you take lowercase dPB and turn it upside down. It spells GBB. And it's another morphogen of the same category. It binds the same kinds of receptors. It turns on phosphomad, but it's expressed more or less everywhere except that it's fed back upon by one of the DPP target genes. So it's not exactly uniform either. And then it turns on some of the patterning genes go right back and feed back upon the production of hedgehog. And then there's this other molecule I'll talk about at the end called pentagon, so named because the fifth vein is gone in the mutant, and so pentagon. And it's turned off by DPP, but it feeds back and affects the range of DPP, probably by inhibiting the DPP receptor interaction. Okay, so it's a big mess, right? This is not unique to the DPP system. Here's the wingless system. Same thing, wingless, receptor, looks nice, turns on target genes turn on other target genes, all looks pretty, but then it turns out that the receptor is under feedback control, there's a proteoglycan, it's under feedback control, degradation is under feedback control, the proteoglycan affects the degradation of wingless, right? There are other targets, and those targets then together affect both growth, which we'll talk about later, and this other target, which then turns on itself. Okay, so the bottom line is this is what you see in every morphogen gradient. It may look like an exponential, but under the hood, you see this kind of craziness. Loads and loads of feedback loops, right? And the question is why? Right? Because think about it. How did we set up the whole morphogen gradient problem at the beginning? Right? The trick was make the morphogen gradient. The cells are stupid, right? They can just say, okay, I'm programmed to turn on this gene at that location. Right? Why would I need all this stuff? Just to make an exponential gradient, I just have to make it, let it fly, and let it be degraded at a constant rate, right? All this is telling us that we've somehow framed the problem wrong, right? We don't really understand what this tissue, what this organism is trying to do with its morphogen gradients, or else it wouldn't be doing it this way. Okay. So this relates to a general point, right? If you're trying to understand the why something is the way it is, that selection is determined by performance, not phenotype, okay? What do I mean by that? I mean, phenotype is what you measure. You bring your, you put on your goggles and you see the system in a certain way and you say, that's what it is. The organism couldn't care less about that. It cares about the role it plays in the physiology of the organism. Does it do a good job? Does it do a bad job? Well, if you don't know what the job is, right, you're not really in a position to make those kinds of judgments. What are some of these performance issues that we often don't think about? They're the kinds of issues that engineers think about. Robustness, noise tolerance, scaling, interoperability. What do I mean by that, right? That the wing is not floating in space. It's attached to the rest of the organism, right? It has to work with those same patterning systems that are happening during development. And speed, how long the system has to get the job done, right? All of those are critical objectives. And so the question is, does all that messiness relate to this, as opposed to just relating to conveying the positional information to this cell? Okay, so we're just going to talk about some of these issues. For example, robustness. It just so happens that wing disk patterning, and in fact, most patterning, not all, but most, is insensitive to loss of one copy of the genes for any of the morphogens, or any of the receptors, or any of the signaling molecules, right? Remember, we're diploids. That's, we have two copies of everything. You can take one away for almost everything, and patterns come out okay, right? Now, that's something, it's easy to understand how maybe metabolism comes out okay. But patterns are supposed to be, you do this at that position because of the amount of that stuff. And now I'm telling you I can take away half the machinery to make that stuff, and the pattern still comes out at the same position. But that's the facts, right? So that's what you see in most patterning systems, a tremendous robustness. Okay, how can we be quantitative about robustness? Well, within the context of the simple model of the morphogen gradient, the simple exponential decay model, right? Well, you can ask, what would happen if I increase the amount of morphogen by a factor of 2 or something? How much does it move over? Well, anybody can do that calculation for an exponential, right? The distance it shifts is proportional to the logarithm of the fold change in the up and down perturbation, right? So for any fold change in the amount of morphogen, every threshold position where you might turn on a gene moves over by a fixed amount, delta x. And the proportionality constant there is that length scale, right? Okay, two-fold change in morphogen should shift everything by lambda log 2. Okay. Now, the problem is, this is great from a kind of physics point of view, but from the organism's point of view, this delta x is not the same as that delta x. Because the organism cares about relative pattern, right? If I'm trying to put something at 10 microns and I put it at 20, that is a much worse error than if I want to put something at 80 microns and instead put it at 90. Okay? The organism cares about the sort of fold change in position, not the absolute change in position. And so a much more useful way of thinking about the robustness of pattern is to use what engineers use, which is the sensitivity coefficient, right? It's just the fold change in output relative to the fold change in input. And it has the nice feature that it's a unitless number, right? 1 is just another way of saying proportional or linear. A half means, goes with the square root. 2 means goes with the square, right? It's a very useful number to talk about the sort of quality of effects of perturbations. And so we're going to use this sensitivity coefficient. Okay. So using that, I'm now showing you a plot of that sensitivity coefficient as a function of position along this dotted line morphogen gradient, assuming the simple exponential model, for different kinds of perturbations. Here we're perturbing the amount of morphogen, and here we're perturbing the amount of receptor. And what you can see is they're different at different locations, right? In the case of perturbing the amount of morphogen, it's a constant shift, but as a proportion of position, it's going down and down over time. In the case of receptor, it goes down, there's actually a crossover point, and then it comes back up again. This is the absolute sensitivity. It's actually going negative here. Okay. So we can then define robustness as having sensitivity below some threshold. 0.3 is kind of a useful one. It means two-fold change gives a 2 to the 0.3-fold change, which is pretty small. This is what engineers would probably be comfortable with. All right. So what do you see here? Whoops. How did I? Okay. So what you see here is there is no region in this gradient where you can simultaneously be adequately robust to a morphogen perturbation and adequately robust to a receptor perturbation. Okay? So you can't do both in this simple exponential model. So then the question is, okay, well, can we use some of this craziness to make it better? Make that go away. So for example, you have this other thing that binds DPP, and it can lead to degradation of the DPP. What if we put that in, let it degrade the DPP, see if we shift things around? Sure enough, voila, I can take now the curves for sensitivity to morphogen and receptor, make them overlap, and have this nice region here where I can be robust to both. What's the problem? No engineers in the group. The problem is I've added a new component. I have to be robust to that, and I'm not, right? If this varies by a factor of 2, right, the sensitivity coefficient is very high. I've essentially taken the fragility from one thing and I've shifted it onto another. That doesn't really solve the problem. Mm-hmm. Because you can't. You're not. So, I mean, that's just the nature of this exponential gradient, is if you shift it in certain ways, the fold change in where you would put a threshold is not constant. It varies with distance. And so the best you can do is make a little region of it adequate. Okay? And you could, in principle, just put that region wherever you want by changing the length scale, but you have another problem, and that's the noise. Right? You have big noise in this system. Not only do you have, you've heard more about this noise here, which is transcriptional translational noise, but you have another kind of noise, which is receptor binding noise. These cells have low numbers of receptors, or low occupancy of receptors. And the receptors are stochastic, right? They're just binding things as a Poisson process, which means if you have 50 occupied receptors on average, this is what you really have, right, over time at that cell. If you have 10, you have right this, okay? So the fewer average occupied receptors, the bigger the noise coming from just receptor occupancy, not even counting all this other kinds of stuff, right? And it turns out that these are very reasonable numbers for receptor occupancy in the sort of far reaches of a morphogen gradient when you're out past, say, past 2 length scales. Now, there's experimental reasons to believe that morphogen gradients indeed have that kind of noise. First of all, I showed you the sort of amount of DPP within cells. You may have noticed how variable it was from cell to cell. But this is a technique that was mentioned earlier today, which is FISH, fluorescence in situ hybridization. This is a slightly different kind of fish. This is called nuclear dot fish. You use a probe which binds to the RNA as it's coming off the DNA. And so since many transcripts are coming off the DNA at once, a lot of probe builds up there. And the fluorescence of the probe is actually proportional to how many transcripts are coming off that piece of DNA. So it gives you the transcriptional rate integrated over some delta t. That depends on where you put the probe, what the delta t is. But it gives you a way to sort of look at the average, look at the transcriptional rate from cell to cell at a constant position within the DPP gradient, and look at its coefficient of variation, which is one way to look at its at the variance, right? And what you see is that it's huge, and this actually has to be corrected down by about that much for noise just in the measurement process, but you can get that under control with 2 different fluorescent probes, and still you're up at close to coefficient of variation of 1, right? Which means the cell next door can have twice the signal that you have. It's really bizarre. How are you supposed to put a stripe somewhere if the cells next door to each other have twice the signal? Essentially, the effect of noise is that instead of having this nice textbook picture of how cells turn on genes at particular thresholds and morphogen gradients, the reality is going to be like that, right? And so the consequence is you get this width, this zone where cells are confused, right? They don't know what to do, and basically it's equal to the coefficient of variation times the length scale. Why the length scale? Because if the gradient's really steep, then the space over which those fluctuations matter is much smaller than if the gradient's very shallow. Yes, I'll show you that, yes. You can indeed time average, but there's a big price you pay for that too. Okay, but I just want to show you biologically that this noise issue is a meaningful one, because there are tricks we can play with mutants that actually make those positional cues noisier. And so instead of making a vein primordium like this, it comes out big and fat. And when it comes out big and fat, what happens is the fly tries to put in extra veins there, because in fact, it's a Turing process that makes the vein at the end of the day. And so if you make the domain too big, you put too many stripes into the process. You get a triple vein instead of a single vein. So the organism cares to keep the noise down. But noise just adds another constraint to this, right? That the region over which you can pattern and be robust and have low enough noise gets even smaller when you consider the noise. Now you can say, wait a minute. How about I just put a lot of morphogen in my system? Just crank the morphogen up, and then I can get the same signal over here, and therefore spread the morphogen gradient even farther. And it turns out you can't do that for another reason, which has to do with the fact that receptors are only human. They saturate, right? In fact, the range over which receptors saturate is pretty narrow, only about 100-fold. And so consequently, oh, this is already running. Okay. I don't know if you can see this. As you add more morphogen into the system, the morphogen may keep going up in a way that looks kind of exponential, but the actual signal cells stops getting exponential at about 30% saturation and starts looking quite different. And if you actually calculate now what happens in terms of the change in position when you have a twofold change in synthesis rate, it's much greater when you're even slightly on the way to saturation. Okay, so what happens is if you try to make the gradient taller to beat the noise over here, then it becomes less robust to any fluctuations over there. And these different kinds of constraints all conspire together so that you basically can't win no matter what you do. And that's actually what this shows, is that as you try to spread the gradient farther, right, to beat one constraint, the other constraint gets worse and worse until eventually it's just impossible to spread the gradient any farther and be robust. And what does this predict? It predicts that there'll be a certain spatial limit over which you can spread morphogen gradients. And beyond that, it's impossible, not because it's physically impossible, as Crick suggested, but it's an engineering problem. You just can't both get rid of the noise and the robustness problems simultaneously. So that gives you kind of an alternate explanation for why gradients reach a maximum size. Okay. How about some other tricks? What else can we do to make these issues of robustness go away? Nama Barkai a number of years ago pointed out that who said degradation has to be linear? What if the morphogen affects its own rate of uptake and degradation, right? Then instead of exponentials, these things will become power laws, right? That's what this solves to. We've now made degradation, essentially the degradation rate constant is proportional to the amount of morphogen signal. And now this solves to a power law. And you can make a higher exponent here, and you get higher exponents there, and so on. But the basic effect of this is that you get something that looks kind of exponential, but if you look close, it isn't, right? These are power law terms. And the nice thing about a power law is if you perturb it up by a certain fold here, it still moves by some constant times the logarithm of 2, but the constant is dependent on the slope here, not the slope here. Whereas here, the constant's dependent on this length scale, which is constant everywhere. And so consequently, you can have something that looks a lot like this in this local region, but shifts a lot less when the gradient is pushed up or down. Okay, that's all they pointed out in their 1993 paper. And so that sounds like a really nice way to make morphogen gradients robust until you think about it for a minute, because remember, I said that the effect of noise on the ability to have precise position varies with the length scale, the local length scale, or the slope at that position. And if you use this self-enhanced decay mechanism, it's steep here because it's very shallow here, and it gets shallower and shallower as you go out. And so consequently, the noise gets worse here than it would be there. And so again, it's a perfect trade-off. You gain robustness here, and you lose it over there. And they basically cancel each other out. And so kind of putting it all together, you have this problem that the cell is trying to read a morphogen signal, and there's many different types of uncertainty that it has to deal with. And every time the cell uses some mechanism to get around one type, it screws up another, right? It ends up having to pay the price somewhere else. And this, of course, is a very common thing in engineering, right? That there are trade-offs, right? You have to pay the price for being robust to one thing or being able to filter something out. So ultimately, this is telling us the cell has an information problem, all right? Because these are independent sorts of perturbations, right? And in order to be able to filter them all out without them interfering with each other, you need more information than just one read of the morphogen, one instantaneous read of the morphogen. You need to be able to add information into how you're determining your position. Okay. So in fact, the cell has access to a lot more information. It has access to time history, right? What that morphogen gradient's been doing over time. It has, and in fact, it's been suggested that if cells decoded the morphogen gradient, that is, if they took their readings of position before the gradient reached steady state, that might actually be more robust. And in fact, that's true. Remember this equation for the approach to steady state, right? Before that's reached, you can calculate what's the change in threshold position. And in fact, it's less than it would be at steady state. Can anybody guess what the price you pay for this strategy is? The time that you measure is critical. If you make an error in when you measure, you can have a much bigger error than you would have had by letting it go to steady state and measure it then. Okay. What else can you do? You can do time integration, as you suggested, right? If you take a noisy input and you use a leaky integrator to just integrate it over different amounts of time, of course you can iron out that noise, right? What's the price you pay for that? Delays. How long does it take to do the integration, right? So here's a case in which, here's your probability of being on or off, right, because of noise in these cells, and that defines a sort of imprecision, a precision width. And sure, you can integrate over 4 characteristic noise times. What do I mean by that? That's kind of noise decorrelation times. Noise has a temporal element to it. So you can integrate for 4 of those times and cut the precision down by square root of 4, or improve the precision, cut the imprecision down by square root of 4. But it takes at least 8 noise characteristic times before the measurement becomes pretty accurate, right? Because you're having to build up the signal over time. So there's a trade-off there. Okay, so the problem then is you can only overcome fast noise, not slow noise. But it turns out a lot of the noise in these systems is slow because the receptors are very slow in that sort of binding and turning over sense. It turns out the receptors have to be slow because they affect the length scale. And in other words, they have to be very slow to bind or else the morphogen can never diffuse very far. So again, you get a trade-off there. Anyway, the trade-offs keep going. You can use the time history to use another strategy called noise-induced switching. Is anyone familiar with that? Okay, so this is a strategy in which you have a bistable system, okay? And if you originally throw points, in a Gaussian of inputs, and they can randomly land either on the top or the bottom branch of your bistable curve, right, then you get a certain variance associated with that. But in a bistable system with noise, there are transitions that are going back and forth, and ultimately, the rates of transition from high to low and low to high are not the same. Right? So you'll end up with a predominant flow towards one or the other of the 2 solutions. And so consequently, the variance will cut itself down over time. Right? And so that's a neat trick, right, for getting a noisy signal to become less noisy by using the noise. And here's an example of And here these times are the characteristic noise times themselves. As the noise changes, it keeps flipping things back and forth before it eventually settles down in where it would be, you know, at sort of thermodynamic equilibrium. And so you end up with a sharper signal. So that's another example. And that's kind of nice because you don't need to know how long to integrate over. Okay, another example is using receptor dynamics to take advantage of time information in the noise. And that actually turns out to be what goes on in the hedgehog signaling pathway. So remember, hedgehog is produced on one end of the disk and makes a nice short gradient. The reason it makes a short gradient is because it turns on its own receptor, okay, patched. And then the receptor eats up the hedgehog. And so you get a very, very short signal. Well, as Marcos Namad and Angela Stathopoulos showed a few years ago, if you model that, it's a little bit ugly, but it's very easy to understand, right? That basically the hedgehog signal is very high until the receptors fall below some threshold at which you can no longer turn on more receptor synthesis, and then it plummets because now receptor synthesis goes down practically to zero. So you essentially just get one switch-like threshold out of this morphogen gradient, which is a puzzle because it turns on targets at 3 different, or at least 2 different positions. How does it do that with effectively one threshold? The key is in the dynamics, that early on in the life of that gradient, it spreads farther than it does later because it hasn't turned on patched yet. And so it flows out and pulls itself back, just like a wave coming out on the shore, right? And then it pulls itself back in to a closer in position. And so it turns out if you have some targets that turn on and stay on if they ever saw a hedgehog, and other targets that turn on but only stay on if they continue seeing hedgehog, then you actually build 2 patterning thresholds out of this. And there's a lot of data I'm not telling you about that sort of support that view that you're using the dynamics. Probably the best example of the dynamics being used to create precise pattern comes back in the bicoid system. Okay, so remember we laid out the bicoid system as just this nice exponential, cells know what to do, turn on stripes and so on. But really what's going on is downstream of bicoid is this huge array of cross-inhibitory transcription factors, each of which is also diffusing at the same time. And in addition, there's at least 2 other morphogen gradients feeding into this, one from the back end of the fly, one maternal hunchback being present in the middle of the fly. And all together, collaboratively, it creates a system where the thresholds, the spatial precision with which these stripes are made, far exceeds what you would expect from what you can get out of a single morphogen gradient. And the person's work, or I should say the people's work, because it's a group of folks, including John Reinitz and Maria Samsonova, the work that's really picked this apart using beautiful quantitative data has sort of mapped this as a series of attractor states, dynamic attractor states, that basically at each point in the embryo, you can follow it as undergoing transitions from one attractor to another. And one of the beautiful things they were able to point out in this paper is there's parts of the embryo where the system doesn't even reach steady state. It actually gets on an attractor ridge and sort of surfs along this ridge for a while until something else happens that shuts the whole system down and starts the embryo along another path. So what's interesting about this view of patterning is even though this is bicoid in its long-range patterning, ultimately this sort of self-organization of attractor states in space smells a lot like Turing patterning. And so this arbitrary separation between long-range and self-organized Turing pattern is beginning to fade away in systems like the bicoid system. This is the level of bicoid. This is the level of caudal, which is another morphogen. And then there's a third axis, which is hunchback, which is not shown here because it's been projected onto 2 so that you could print it. But there is actually a third one there. And then these map out the attractor landscape. And then these are experimental points, and I'm not exactly sure. I'm not sure I remember where they come from.
What does it mean that bicoid only does all the exciting things up to n, and then after that?
Well, this would be close to the bicoid source, high concentration. And this would be low. So it's just saying if you didn't have these other things, everyone would be in one state, right? But you already, so you tend to have low caudal where bicoid's high and high caudal where bicoid's low. So I think a lot of the business is happening along here. Anyway, I recommend you read the paper. It's a really nice paper, but it's a kind of slow read because you have to go through, they go through step by step through each of the stripes, what the dynamic is at that position in the gradient. I just think it's a great example for what's likely to be the case for a lot of other morphogen gradients. Okay, I only have a little bit of time left, but I think I'll finish. So I just talked about one kind of robustness or one kind of performance issue, which is what we call parametric insensitivity and noise, right? Being insensitive to these things so that your morphogen gradients will be the same, right? But that's not really always what the objective is, because I mentioned earlier on, right, that morphogen gradients have to adapt. If the wing is going to be bigger, you have to make a bigger gradient. So you have these competing objectives of robustness and adaptability, and yet you have to have them both, right? In particular, scaling is really common in pattern formation. I showed you this picture earlier. This is a classic picture from Driesch in the 1890s, that you take a sea urchin embryo and you split it at the 2 cell stages, and each cell makes a half-size sea urchin, and it's perfectly patterned. And this is a frog, where the frog embryo hole patterns like this. You can cut the embryo in half, and the dorsal half will make a half-sized frog. The ventral half won't, but the dorsal half will make a half-sized pattern. So these patterns have an intrinsic ability to scale. And in fact, even the DPP gradient scales. During normal growth, that length scale is actually changing over time up to a point when it levels out about here. Okay. And in addition, there are tricks you can play to make one half of the disk grow larger compared to the other half. And you can see the gradient on just that half scales to match the new size of the disk. Okay. So there are 2 ways to scale. This is fundamentally, either you scale open loop or closed loop, or using feed forward or feedback means. So what do I mean by that? Feed forward means that whatever disturbance is changing your size, that same disturbance is going to be coupled to changing your pattern. And now you're safe, right? So if you're going to starve, you make your pattern smaller as a result of starvation, not as a result of being small, right? The opposite is feedback, right? Where the system is somehow measuring its size and correcting the gradient to fit the size. Some open loop scaling comes for free, and that actually relates to the comment Rob made earlier about the bicoid length scale in different flies. And some of this is just open loop scaling. The geometry of the different size fly actually changes the gradient on its own. But in the DPP system, actually there's some open loop scaling that comes for free, because remember how tall these cells were, right? This is in cross section, right? Well, there's a reason for that, at least we can imagine a reason for that, which is that the morphogen diffusing in the spaces between cells is protected from leaving through the top by things called tight junctions that zip the cells together, but there are no such things at the bottom. And so morphogen can just flow out the bottom. And presumably there's always stuff flowing out the bottom. If I scale this and make it bigger, then the length from here to here gets longer and less flows out the bottom, which means if I make the disk bigger, the decay of the morphogen is less, and the length scale of the morphogen gets greater. So there's kind of an automatic scaling that comes to play just by making the thing bigger. That's an example of open loop or feed-forward scaling. In fact, Wolpert's original idea for scaling was an example of open loop, right? You just move this around, and automatically the gradient follows, right? This is just a, where'd my mouse go? All right, that's just an example. But as Boris pointed out, this doesn't actually work. There's 2 reasons it doesn't work. One of which, of course, is gradients aren't linear, right? They're exponential. And what you see with the exponential gradient is it will only scale by moving a boundary position up to about 1 length scale, and then the boundary position has no effect on the shape of the gradient anymore. The other reason is Wolpert had to assume that the source was pinned, right? If you do the math without the source being pinned, if you say there's a real source there producing at a certain rate and you move the sink, it doesn't scale. This is going up the whole time, right? And that's because the removal from the source in the source sink model depends upon the sink. If you have real production here and you move the sink, you're removing less from it, so it has to go up. Okay, so it actually never worked, even though that's what was in the paper. It's just the math doesn't work for that. Now, in the case of the DPP gradient, there's recent strong evidence that this strange thing called pentagon actually plays a role in scaling the gradient. So pentagon is a secreted molecule. It inhibits DPP, probably just by blocking DPP binding to the receptor, but its expression is repressed by DPP. Okay, so here, when you take pentagon away, the DPP gradient shrinks. So think of it as a length scale controller for the DPP gradient. But pentagon is only expressed out here where the DPP gradient isn't. All right? And this fits a model that Nama Barkai put forward a few years ago called the expander repressor integral feedback control model. We'll talk tomorrow about integral feedback. I don't have too much time to talk about it. But basically, the sort of fundamental thing in this model is that you have something that can expand your gradient, but it's produced where the gradient isn't. Okay, so if the gradient expands, it lowers the production of that, which then de-expands or contracts your gradient. Okay, it's integral feedback because in the model, this substance will accumulate forever unless you suppress it more or less completely. So essentially, it doesn't turn over and it diffuses everywhere, which is how it can control the length scale of the gradient. And it turns out many things about pentagon fit that model, right? And so a couple of papers came out saying, yep, these are these scaling plots where you look at the gradient normalized to its own size. You show that you mess up scaling if you get rid of pentagon. But unfortunately, it's one of those cases of a great model that is kind of slain by a bunch of ugly facts. The model depends on the idea that pentagon diffuses everywhere, right? And its concentration just reflects time and the size of the system. But the problem is, remember I said everything leaks out the basal part of the cell. So pentagon can't have essentially an infinite length scale. It can only go so far. The second thing is, I mentioned that the anterior and posterior halves of the disk scale independently of each other. You can make one scale and keep the other the same size. And that's inconsistent with the idea that pentagon is just spreading everywhere. And then the third thing is one of my students has now measured the length scale of pentagon itself using fluorescent pentagon, and it's only about 13 microns. Remember that each compartment of the disk is like 80 microns wide. So pentagon doesn't actually diffuse that far. So how does it work? The first clue is that the pentagon domain is actually where receptors are expressed most highly. Remember I said the receptors aren't uniform? The receptors go up towards the edges because they're repressed by DPP. Is it possible that pentagon in the periphery affects receptors in the periphery and somehow that decreases the length scale globally? All right? And it turns out that actually works. So this is on a true axis and the bottom one's on a scaled axis. So this is the case in which pentagon's only acting out here, right? But I can get the gradient to scale just by affecting those receptors out there. In other words, it gives you a reason why you would want the receptors to be concentrated on the outside, because then by controlling them, you can control the length scale of the gradient on the inside. Okay. With one caveat, it suffers from that same problem that I brought up with the Wolpert scaling, which is that I'm assuming that I've pinned the concentration there. That's not a realistic assumption. However, this is something I didn't have time to show you. The fact that DPP gradients are actually very robust to the width of that region suggests that there's a very strong capture of DPP within its own production region. In other words, it has a very short length scale within that region, and that would effectively pin its concentration. But that can't be due to receptors because they're almost absent in the DPP production region. And yet, we can show that the length scale is short. And this is a bit complicated, but this is looking at, we forced DPP GFP to be expressed in this direction instead of that direction, so we can sort of probe the length scale at different places. Now, this is, you can't really use this to measure the length scale because there's so few receptors here that you couldn't trust these numbers anyway. But what you can trust is the effect of this length scale on the length scale nearby. And let me just show you what that looks like in modeling. Right? If there weren't much DPP capture here, so that there was a really long length scale there, you would expect the nearby gradients to bend up towards it. Whereas if there's a really short length scale, you'd expect the gradients to bend down towards it. If it's the same length scale, you'd expect the gradients not to be perturbed. I think you can see that that's what's the case here. This gradient is kind of flat. This one's clearly bending up away from that. So that suggests the length scale in here is about 15 microns, similar to the length scale over here. And that's probably due to the fact that that proteoglycan that I showed you earlier is expressed in that region, because if we up its numbers, we can actually keep very much DPP from getting out of its production region at all. And so consequently, when you put all that together, this short length scale of DPP in the production region gives Pentagon the ability, together with the repressed receptors that push the receptors far out, to actually produce that scaling behavior. It also produces the ability of DPP gradients to be insensitive to the size of their own production region, which makes them then insensitive to hedgehog. And then there's additional robustness to hedgehog dose for another reason, which is that DALI actually plays a role in hedgehog signaling. So when you put it all together, we can explain potentially some of this stuff. And even in some cases, if we're wrong, right, still conceptually, we're saying, you know what, whatever's going on, you're going to need a lot of complexity to deal with these performance objectives that the real organism has to deal with. And we may still be missing quite a lot of it, and there's still a lot here that remains to be explained. But the simple idea that we can just kind of take the morphogen gradient and think about it as an exponential and just sort of treat it at that level leads us very far astray if we try to run with that. Okay, so what's my summary? Patterning systems aren't just systems that pattern, right? It's not about phenotype, it's about performance, right? And there are many performance objectives, including accuracy, precision, robustness, size, speed, scaling, and trade-offs abound. It's just like in engineering, right? You can't make it perfect for one thing without screwing up something else. And it's this issue of balancing among these different performance objectives that gives you the constraints that force the system into this very high level of complexity and kind of maps out for us what it is we really need to understand. Okay, so, all right, roughly on time. Anyway, stuff I told you about, some of it from the literature. I hope I cited everybody correctly. In my lab, the folks doing the work I talked about are Abed, Marcos, Peng, and Jimmy. Xiaohua did the beautiful work on fluorescence correlation spectroscopy and photoactivation in DPP. And these are some of my math collaborators who've been really important in this. Okay, thanks a lot, and I'll take any questions you have. Yes?
Say an organism is growing, is the scaling perfect at every given instance, or are there regulatory mechanisms that sort of correct for errors that might be made so that the finished product is?
The scaling is not perfect. There's a nice paper by Fiesenhammer-Tolu that actually goes threshold by threshold for the different genes that are turned on and looks at the quality of scaling in the anterior and the posterior compartments. And it's definitely not perfect, which is good because the expander repressor model predicts it should be perfect, whereas the other models like the ones we're working with predict it won't be perfect, and it gets less perfect over time. But the next part of your question is, are there things that come in later? Yes, there are, and most of them are really not very well understood. There seem to be things that help kind of even out the spacing between veins. at a very late stage in larval development. And that's still very poorly understood. What are these veins for? Yeah. So there's a couple things. The main thing is probably mechanical struts, right? If you look at like old balsa wood airplanes and stuff, right, you have to have these struts in order to keep the thing stiff, because if you think of the speed at which these things beat at. So stiffening is probably one of the main functions, but in addition, these things are hollow, and the sort of immune cells or blood cells, if you will, of the fly actually go through these hollow tubes and use that to disperse in the tissue.
Another question is how, even if there is noise, the scaffolding overall works, does noise really matter in this particular case?
Well, so one of the reasons noise matters, I didn't have time to explain this, is, so remember I said the disc has an anterior half and a posterior half and a dorsal and a ventral, right? I sort of drew it as though the veins on the disc are the veins of the wing, but actually the disc does this strange thing that in pupil life it folds down the middle, and the veins on the dorsal half and the ventral half have to meet each other in perfect register in order to form the final veins. And so if that's screwed up, you get and a lot of the phenotypes that you see are because that registration doesn't work. The other thing is the sharpening of those veins into thin lines is actually achieved by a Turing process that's on a very short length scale. So it basically makes one stripe. And if you get the specification of the vein primordium too wide, you make 2 stripes or 3 stripes. And so you get extra veins. And they kind of start and stop, and they're not connected. And so probably mechanically they're pretty bad. In the case of camouflage, sudden changes. So you're talking about pigmentation patterns. Well, so usually those kinds of camouflage, short-term camouflage changes aren't changes in the fates of cells. The cells are there, right? They just change their behavior momentarily to either sort of retract a pigment or into the center of the cell or something like that. But here we're talking about the sort of early events in development that tell cells what fates to adopt, sort of what kind of cell to be at what location. But after that, there are physiological things they can do that change the sort of visible coloration patterns in some organisms.
Actually, I think we may have to
Yeah.
dedicate this
Okay.
today because we have to go to ICTP-ICTS and hopefully wait till the meeting to take us there.
Okay.
Thanks very much.
Thank you.